The next topic it is the applications of DNA fingerprinting technology. What are the applications of DNA fingerprinting technology? First one is it's used to identify criminals and rapists. Second, to solve parental dispute. Third, to solve immigrant problems. Fourth, to identify dead bodies of soldiers died in wars. Fifth, to identify dead bodies of persons died at accidents and bomb blast. Six, to identify racial groups. Seven, to detect inheritable disorders. Next point, to detect donor cell in case of transplantation. So in all these fields, this DNA fingerprinting technology, it is very applicable. We are using all this DNA fingerprinting technology. So it is very useful in our day-to-day life. So these are the applications of DNA fingerprinting. So it is used to identify criminals and rapists, to solve parental dispute, to solve immigrant problems, to identify dead bodies of soldiers died in war, to identify dead bodies of persons died at accidents and bomb blasts, to identify racial groups, to detect inheritable disorders, to detect donor cell in case of transplantation. So it is very useful in our day-to-day life. Then what are the steps how the DNA fingerprinting it will do? The DNA is isolated from the sample of blood cells, hair root cells, semen or bone collected at crime spot. So first there will be DNA is isolated from the sample of blood cells or hair root cells or semen or bone collected at crime spot. Second, what they will do, the DNA of suspect also collected and isolated separately. So they got DNA from one place, from the crime place, and at the same time, the DNA of suspect also collected and isolated separately. The isolated DNA is treated with REN, renin precursor, to cut into number of fragments. So this isolated DNA is treated with REN, means renin precursor, to cut into number of fragments. The DNA fragments are separated according to their length on gel slab using gel electrophoresis. How these fragments are separated? These DNA fragments are separated according to their length on gel slab using gel electrophoresis. I told before what is meant by electrophoresis. The movement of charged particle in a fluid or gel under the influence of an electric field, that is called electrophoresis. The DNA strand now they separated. After that, the DNA strand on gel slab is treated with alkaline solution to split double strand into single strand. The next is this double stranded DNA we have to split into single strand. For that, what we will do, this DNA strand we have treated with alkaline solution to split double strand into single strand. The single strand DNA is transferred to nylon sheath using Southern blotting technology. Before you learn what is meant by Southern blotting. So the single strand DNA is transferred to nylon sheath using Southern blotting technology. The single stranded DNA is hybridized with radioactive probes of VNTRS. The excess of probes are washed off. Okay, so the next step is single stranded DNA is hybridized with radioactive probes of VNTRS. The excess of probes are washed off. The next, nylon sheath is X-ray photographed to get bands of VNTRs. They are X-ray photographed, so we will get the bands of VNTRs. The bands of X-ray sheath is the DNA fingerprinting. So this band of X-ray sheath is the DNA fingerprint. Then after getting this fingerprint, comparing the DNA fingerprint of sample collected at crime spot with suspect identifies the individuals. So what they will do, they will be doing comparison between these two. Which one? The collected from the crime spot and one the suspected persons. So the comparing the DNA fingerprint of sample collected at crime spot with suspect identifies the individuality. Then what is Southern blotting? already we told, the technique of transferring DNA from agar gel to nylon sheath is called southern blotting. What is PROP? Single-stranded polynucleotide fragment complementary to specific sequence of nucleotides of DNA is called PROP. Single-stranded polynucleotide fragment complementary to specific sequence of nucleotides of DNA. It is called PROP. It is mainly used in identify VNTRS and desired gene. So we have completed this chapter 6, molecular basis of inheritance. The last topic it was HGP, Human Genome Project, and DNA fingerprinting. So in the next class we can start the next lesson. So if any doubts, children, mail your queries on dmcsonlinequeries@gmail.com. Thank you, students. Sure.